I did keep the desk spread out. I don't know, did you guys hear about the different rules? So we'll try and kind of keep the desk 3 feet apart to keep with that when possible. So we're going to start our next section today, which is really still Chapter 8, but just the second part of Chapter 8 in your textbook. And so this is hopefully a quick question, but go ahead and talk about that with your group real quick to get started. Yeah, so still work with the same people you normally talk to.
Got me some.
So those A, T, C's, and G's, that code is eventually going to get used to make. Well, we can make more DNA. Yeah, we can do that. So we can copy the instructions and make more DNA. But eventually, the reason why every cell needs that information is that we're going to use it to make proteins. Yeah. And you may or may not remember, but proteins basically do all the important functions in your cells and therefore in your bodies. So this whole next part of the chapter and what our next test is going to be over is how do we get from that DNA sequence, A, T, C, G, A, whatever, to proteins. So that's the whole point of this next section. Go ahead and pull your folders out. We're going to be looking in there today. The next couple pages. So before we get into that process of how do we make proteins from DNA, we're just going to go back and look a little bit on proteins in general. So this next section in our folder at the bottom, it says Chapter 8, Part 2. So this is, this is the second part of Chapter 8, and it will start over at page 1. So we're going to do page 1 and 2 today, and then there's a Canvas quiz assignment over that material. This basically is like goes through all the material in these 2 pages again to make sure that you have everything you need to know. So if you read the little info blurb at the top there, it says DNA holds the information for making proteins, which is what this next unit is all about. Before we learn how to make proteins from the information in DNA, we're going to review a little bit about protein structure and function. So there's a video we're going to watch. I think we might as well just do this part all together. So we'll watch the video together and kind of pause it when we hit points that we need to discuss. So some of this, hopefully, you kind of looked at last semester, but if not a big deal. So hopefully, let's go talk about more. I'm having a lot of issues with my. Okay, we'll have to just look at the slideshow. So, sorry. All right, so first question says, what is the building block or monomer of proteins? Those are called amino acids. So if you remember that term amino acids, hopefully from last semester. So there's 21 different amino acids out there that we can use to build proteins. So we can build a chain out of any sequence of these amino acids. So you can see there's a wide variety of like size and shape here, right? When we look at a protein, we're going to see the same atoms multiple times. So the atoms that we'll see in proteins, the atoms that proteins are made up of are carbon, oxygen, nitrogen, hydrogen, and sulfur. So it's okay if you don't remember this, but in the Hershey and Chase experiment with the viruses, we grew some of those viruses in radioactive sulfur, right? So that radioactive sulfur is what we use to tag the protein. Those are the building blocks. When we look at those 21 different amino acids, they actually have a very similar structure except for one part. So all of them are going to have this what we call the central or alpha carbon. They all have this what we call a carboxyl group, which is what makes it an acid. They all have this amino group, but what's different about them is what we call this side chain. And so what the question says, what part of an amino acid is different from each other? It's this side chain. A lot of sources will also call this the R group. Okay, so that's one of the questions in the Canvas quiz later too, is the side chain of the R group. So again, that's what's different. For each one of those 21 amino acids, there's different side chains. So they all have the same alpha carbon, they all have the same carboxyl group, the same amino group, but the side chain is different. So what's important about that side chain is that based on what atoms are in it, you're going to have very different properties. So really quickly, hopefully these terms are somewhat familiar. Hydrophobic, do you remember what hydrophobic means? What's a phobia? Yeah, something you're afraid of, and hydro refers to water. So these are molecules that are going to be repelled by water. Yeah, hydrophobic. These are hydrophilic. Philia means to love. Okay, so these are water loving. They're going to be attracted to water molecules. If you remember the word polar at all, so remember water is polar, it has negative oxygen and positive hydrogen ends. So hydrophilic molecules are also polar. They form attractions with an opposite charge molecule. Some of them have actually positive or negative charges. It also makes them hydrophilic or attracted to water. And so you can see, a good chunk are hydrophobic. The rest of them are hydrophilic or charged, which also makes them dissolve well in water or attract to water. And so based on the combination of those in your protein, parts of your protein are going to not want to be close to water, so they're going to huddle together on the inside. Parts of the amino acid of the protein are going to love water. They're going to want to be on the outside of the protein in the cytoplasm where it's all water. And so that's kind of what it's going to show here on the next couple slides. So here is a R group or a side chain that's hydrophobic. Notice all the water molecules are kind of not attracted to it, right? So they don't interact well with water. Here's the hydrophilic side chain or R group. It forms those hydrogen bonds with water, right? It interacts really well with water. Some of them are charged, which not only are going to attract to water, but are also going to attract really well to each other, because positive and negative charges do what? Attract. So we'll get to this later in this unit, but every 3 bases in DNA are eventually going to code for 1 amino acid, and we bond them together. Let's see the next. And so we have these different levels of protein structure. So it does ask you to write a little bit about each one of those in your notes. So here's primary structure. Primary structure is just a series of amino acids. So this says a linked series of carbon, nitrogen, and oxygen atoms, but I would just say a chain of amino acids linked together. And depending on what the DNA sequence was, the amino acid order is going to be different too. So every protein has a unique order of amino acids, has a unique primary structure. Then we get to secondary structure. So this is a folding of regions of a polypeptide chain. And there's really only 2 types of secondary structures that you'll see. You'll see these helices, alpha helixes, something called beta sheets, beta pleated sheets. So this is just within a single region of the primary structure. Sorry, I wish that video was working. It was a lot more exciting. The animations are helpful. But this is just more on the secondary structure. So here's again the helix. So that those forms because of hydrogen bonds between the amino acids, and here's an example of the sheet. So again, you can see these hydrogen bonds that are forming between amino acids in the chain. So those are secondary structures because you can see where we have some folding of that primary amino acid strand, right? So that's secondary. Tertiary structure is where it gets pretty complicated, and this tertiary structure depends on the properties of the side chain. So the hydrophobic amino acids are going to want to be on the inside, and the charged and hydrophilic ones are going to want to be on the outside. So this is the 3-dimensional shape of the protein chain. And it's really determined by the characteristics of whether you have hydrophobic or hydrophilic or charged side chains, in what order they come in. Sorry, I know this font is probably small for some. So I don't know how well you can see this, but can you see a helix? There's a helix here, and you can see a beta pleated sheet back here. So those secondary structures you're going to see within this tertiary structure. Okay, so this is just another folding on top of the folding. So here's a couple examples of what proteins look like. So here's one tertiary shape. Here's another tertiary shape. So that tertiary level is what really gives unique proteins their unique structures. So all proteins have helices or pleated sheets, but every individual protein has a unique tertiary structure, and that allows it to do a particular function. Okay, so there's hemoglobin. Quaternary structure is then when you have two or more tertiary structures that join together. So two or more polypeptide chains that come together are going to form a larger quaternary structure. So hemoglobin is the example here that is four of those tertiary folded polypeptide chains that come together to form one quaternary hemoglobin. This is just a couple different ways that we represent proteins. So you don't have to go anything in your notes about that. Tell you a little bit about the size of proteins. So some proteins are really, really small because they have to fit inside of your blood cell. Some proteins are really, really big. And their shape determines their function. So the very last question here says 6 different functions of proteins. So at the top here, it's going to talk about some of its functions. One of those functions is defense. So antibodies, for example, have the special arms on them that are going to detect different substances that are foreign. So it might detect a certain bacteria or a certain virus. They're important for communication, so that's the next function. Anybody heard of insulin before? So insulin, if you're diabetic, you have to take insulin because your body doesn't make it or respond to it properly, depending on the type of diabetes you have. And so insulin is really important to maintain that blood glucose level. Enzymes, we talked a lot about enzymes. They either put things together or break them apart, right? They cause chemical reactions to happen. Transport. So last semester we talked about facilitated diffusion and active transport, which require transport proteins, so channel proteins or pumps that help things get in or out of the cell. So here's an example of a calcium channel. Storage. So some proteins are just responsible for storing things. So this example, ferritin stores iron. And the last function is structure. So all your cytoskeletons made out of protein, collagen fibers, these are what make your skin elastic, right? So all kinds of proteins that help to actually build your structures inside your cell and therefore in your tissues and your body. So that was what we were supposed to get from the video. So again, I apologize that didn't work. I'll try and link this PowerPoint too if you need to look back at it later. On the back side, on the next page, there's a second site that everybody's going to go to, and I hope this one works properly. Did yesterday. Go ahead and get your Chromebooks out. And if you go to today's agenda, obviously that first link doesn't work. I'll have to fix that. But link 2 is the one you're going to use to fill in this back side. It's called Molecular Machinery, a tour of the protein data bank. So if you click on that, it looks like this. Not every molecule on here is a protein, but there is a bunch of different proteins here that do all kinds of different things. So I just want to point out a couple things here. So this line that runs along the screen here, if you look at the bottom, notice it's color-coded purple. This is a cell membrane. So these are all proteins and molecules that are part of the cell membrane. The blue to the right side, these are all proteins and molecules that would be inside of the cell or in the cytosol. On the very right, the orange molecule is actually DNA. And so those are all proteins that are associated with DNA. On the other side of the membrane, to the left-hand side of the screen, those are all extracellular proteins, so proteins that are outside the cell membrane that might be like in your bloodstream and stuff like that. So there's a table here in your notebook of different protein names. There's a picture next to their structure, what they look like. What you'll have to do is find those proteins, and I'm just going to pick a random protein here, but when you click on it, and this is running a little slow, but it should load a couple representations of the protein, but it should also tell you on the left-hand side what its function is. Okay, so you'll have to click around and look at these different proteins and figure out and fill in what their functions are. So that's your job for the next 10 or 15 minutes or so, however long it takes you to fill that in.